Thank you very much. So what I would like to do today is talk a little bit about how we take a field of science that really is about observation of organisms under different conditions and transition this very fundamental natural science type discovery of a biological system into treatment for disease. For many, I should have practiced. So for about 25 years now, my laboratory and I have been interested in a natural science phenomenon. And that natural science phenomenon is called circadian rhythms. It comes from the idea that all organisms evolved on our planet, and they evolved to adapt to a planet that experiences cycles of light and darkness. Oh yeah, don't worry. Thank you. Excuse me. So these cycles of light and darkness have caused many physiological and behavioral rhythms to evolve in almost all organisms that we know on the planet. Of course, we know this as humans, for example, when we travel across time zones and we experience jet lag. So right now I'm standing in front of you and my brain thinks it's in Los Angeles, but really my body is here in Guiyang. And that dislocation, or that lack of harmony between my internal circadian program, which is about genes and proteins, and the external world, which is about cycles of light and darkness, or warm and cool, that disharmony or desynchronization is what's making me feel a little less than optimal in my performance right now. So this whole field starts by observing organisms, looking at how those organisms have adapted to their environment, and trying to ask some simple questions. That is, how does an organism actually have an internal biological clock? If you were given 20,000 human genes and told to build a biological clock that would allow an organism to take advantage of living on a world that spins on its axis, how would you build that clock? Which components would you choose? How would you make that clock relevant and to help that organism adapt more effectively? And in the United States, as I talk to friends of mine who practice acupuncture and qigong and other fields like this, I began to learn that there were elements, for example, of traditional Chinese medicine that were a little similar to some of the questions that we were thinking about in my lab. Those elements, for example, are the idea in traditional Chinese medicine and philosophy of the idea of how different organs are activated at different times of the day as a consequence of the flow of qi through the acupuncture channels as a function of time throughout the day-night cycle. And my friends who practice TCM in the United States would tell me about the concept of harmonizing habits. That is how, if we understand how our organs have peak functions at different times of the day, we can develop harmonizing habits in order for us to live the most healthful type of lifestyle. So I don't know how many of you in the audience today have an iPhone. Raise your hand if you have an iPhone. Not many, maybe. So, for example, studies in modern circadian biology have shown us that exposure to light from iPhones and iPads and Androids is actually quite harmful to the disruption of our sleep-wake cycle. And so that is why, for example, if you had an iPhone, rather than all those Androids that I see out there, you would find that in iOS 9, they introduced a mode into this iPhone called night shift mode. And if you put night shift mode on, it removes the blue light from your screen, because that blue light activates a photoreceptor in your retina that our laboratory was involved in discovering, and that photoreceptor in your retina resets your biological clock every day. So it's about harmonizing habits, harmonizing behaviors that allow us to understand that really we have evolved in the context of a 24-hour light-dark cycle. And a lot of the modern technologies that we're developing are about helping us to combat shift work, jet lag, the use of light-emitting devices that disrupt what would be our natural sleep-wake cycle. So I think what's really necessary here is the concept of medical bilingualism. The idea of being able to share certain philosophies about the approach to understanding natural science and its application to medicine. And in this way, I think, we're going to be able to find, as I said, touch points between these two approaches. This should not be about the battle of histories or the battle of cultures. This should be about the unification of ideas and synthesis and the acceptance of plurality when it comes to treating medicine. And for Westerners, I don't think these books would be very informative to you guys, because you already know so much about this. But for me, two books have helped me understand a little bit about this type of approach. One is by Lei Tiang Lin, who's at the Harvard Yenching Institute, who published this book, Neither Donkey Nor Horse. And the other is by Volker Scheid, who's a professor in London who practices TCM and has written a very interesting book about the role of traditional Chinese medicine in contemporary China. And again, these books have been written, of course, for Western audiences rather than for experts such as yourselves. So I now want to shift from this idea that comes from TCM of the flow of qi and the idea that different organ systems are activated at different times of day to the kinds of questions that myself and my graduate students think about every day in our lab. So we think about the human system, and we think about how, when we think of the timing of the clock, we think about all of the different physiological functions that are known to cycle throughout the 24-hour cycle. We know, for example, that our blood pressure starts to dramatically rise, as does our body temperature, just prior to dawn. We know in the evening, hormones like melatonin, a hormone that signals the need to sleep, rises in our bloodstream at night and promotes sleepfulness. So just as we could think about the activation of different organisms in the concept of qi, modern biomarkers are showing us that's exactly what's happening. We can see that different organs of the body are actually having different functions throughout the day-night cycle, and those functions are controlled by an endogenous 24-hour clock inside of the organism. So over the last 25 years, the job that my lab and our collaborators throughout the world have had is to really understand what these biological clocks are, and how can we then apply that basic scientific knowledge to the development of new medicine. And we have to think about our body being an orchestra of different tissue-specific clocks. The clock that is in our brain that controls our sleep-wake cycle is a little bit different, for example, to the clock that's inside our liver that controls things like glucose homeostasis, gluconeogenesis, lipid metabolism. So our body is really a suite, like an orchestra, in which the master clock, which sits in our brain, taps out the timing signals to the rest of the body so that we can coordinate activity with food intake, that we can regulate our light-dark cycle, and that the organism can optimally adapt to its environment. And as we start to study these clocks at the molecular level, we begin to find a great deal of association between these clocks and between different disease states. So for example, in as humans age, we find that their circadian rhythm becomes weaker. The actual amplitude of that rhythm dampens. And many different aspects of human physiology and biochemistry start to become impaired as a function of the 24-hour cycle with aging. So what we're beginning to discover is that the pathogenesis of diseases of aging, the pathogenesis of chronic diseases that we see emerging in the West, many of those diseases actually have a molecular pathology that lies in these mysterious 24-hour circadian clocks. And what is really interesting to us as we start to identify all the genes that cycle in different organs and the proteins that cycle in different organs, is that if you look at either the World Health Organization list of the 100 most essential drugs, or you go to the FDA and you look at the 100 best-selling drugs in the world, what you find is more than half of the targets of those approved drugs are actually circadian rhythm products. They're actually protein products that cycle with a 24-hour rhythm in a particular target tissue. So we heard a little bit from the previous speaker about the idea of restoration to harmony within the environment. And in a way, a parallel of that philosophy is what we have in our lab. We have an understanding that the circadian rhythm is actually diminished in different disease states. It's diminished in the aging process. And our job, therefore, if we want to apply this basic scientific knowledge to medicine, is to develop drugs that would restore or reset circadian rhythms in different tissues, thereby providing novel therapeutic approaches to a wide range of diseases. And we not only use a lot of technology platforms that allow us to discover new drugs and apply them, we can also use modern genetic platforms to identify those patients who actually may have a defect in their circadian rhythm on a genetic basis. So we have a very big collaboration with the company 23andMe. 23andMe is a company that's been spun out of Google. And 23andMe has accumulated a database of about 1.8 million consented participants for which they've done a great deal of DNA sequencing. And so we're working closely with 23andMe to, for example, we can give questionnaires to those 1.8 million people and ask, do you prefer to wake up early in the morning or late in the morning? Do you prefer to stay up late at night, or do you need to go to bed early? We can start to look for genetic causes of circadian defects, and that's one way in which we can use modern human genetics, what we now call PHEWAS, phenotype association studies, rather than genotype association studies, to actually begin to segment patient populations so that we can do personalized circadian therapy. Clocks are very important if you want to understand cancer and novel treatments to cancer. So for example, WEE1, which is a classical target of cancer, it's a protein that's involved in regulating proliferation. WEE1 cycles with a huge amplitude in the target tissues. And so if we can understand how we can target WEE1 as a circadian rhythm protein, we can begin to deliver benefit to the patients in the clinic for that as a novel cancer treatment. Another treatment that, another cancer that doesn't have many good therapies is acute myeloid leukemia. When we use classical systems biology approaches, that is large-scale genomics, systems genetics, proteomics, together with our collaborators, we've been able to show that circadian clock proteins play a critical role in the differentiation of the stem cells that are responsible for acute myeloid leukemia. So by chemically targeting circadian clock proteins in AML stem cells, we've begun to develop novel treatments for this leukemia that serve a real unmet medical need. The same is true for other cancer situations that I'm going to skip over because of time, but the circadian clock regulates the protein stability of a very important oncogene called MYC. So we now have begun to develop drugs that interfere with the way that circadian clock proteins, like this one we call CRY or cryptochrome, that regulates the protein level of MYC. And we think in that way we're able to control the protein level of MYC in different states of cancer, and that's going to again give us a novel way of treating those cancers. If we think about our lymphocyte system, it's now been shown that the circadian clock regulates egress of lymphocytes from, for example, the spleen and lymph nodes to target sites. So again, drugs that regulate the clock can be used to regulate lymphocyte egress. And lymphocyte egress is going to be important if we want to develop novel treatments, for example, to autoimmune diseases. So diseases like inflammatory bowel disease or multiple sclerosis. These are autoimmune diseases where what we want to do is to develop drugs that tell the lymphocytes to stay in the spleen, to stay in the lymph nodes, and not go and attack your own organs. So again, I want to emphasize, this all started as a field where we could observe organisms having different behaviors, different activities on a 24-hour basis. But as we begin to apply genomics, molecular cellular biology, and genetics, it begins to reveal novel ways in which we can treat disease. Now, of course, the very first applications of understanding the circadian system that comes from these studies is to develop new sleep drugs. So I mentioned to you that there is a hormone that cycles in our body, melatonin. In fact, in the US, you can walk into a health store and buy a bottle of melatonin, and many people tell me they like to take melatonin when they're traveling to conferences and it helps them sleep. Unfortunately, that's a really good example of the placebo effect. And the reason is that in most humans, not all because we're genetically diverse, melatonin has a very short half-life in the stomach. So for most of the people who think that if they take melatonin orally, it's helping them to sleep, are in fact experiencing a placebo effect. And placebo effect actually is very well known in the development of sleep therapy. So what drug companies have done, of course, is to take the natural product, melatonin, it's a natural product after all because humans make it, and they've altered the chemistry to make forms that are stable for oral administration, and those forms now turn out to be very effective sleep drugs. So again, we're quickly moving from understanding a complex biological phenomenon like a clock to developing specific therapeutics for specific types of diseases. In the lab, what we've discovered is that these biological clocks are really a complex molecular circuitry. If you were to open I know most of you don't wear watches anymore if you're under 30 years old because you have phones, but for old people like me who still wear watches, if you open up that watch, you'll find cogs inside of that watch. And those cogs essentially describe a feedback loop. That's how almost all watches work. The quartz crystal inside of modern watches do the same thing. They create an oscillation. So inside the cells of our body, we have exactly the same kind of mechanism. We have a complex genetic circuitry inside almost each cell of our body that creates a 24-hour circadian rhythm that regulates gene expression and protein level. And one of those proteins, which is called cryptochrome or CRY, has turned out to be a very important protein in terms of understanding certain disease states. We published a paper in Cell recently together with our collaborator Ron Evans that showed this complex molecular circuitry tends to break down, for example, I'll stop doing that, tends to break down, for example, in metabolic disease. So we now find that in diabetic patients or patients that have dyslipidemia, the amplitude of the circadian clock becomes reduced because some of these regulatory proteins that we study have been altered in their levels by the disease. Again, this is showing a connection between having a weak circadian clock and having then the susceptibility to develop a chronic disease. So what kind of drugs can we make that take a weak clock and make it stronger? This protein, cry or cryptochrome, is now becoming implicated in a number of different key diseases. And I'm sorry that there wasn't time here to translate these slides into Mandarin for you. But basically, I just want you to understand that just in the last few years, we've discovered that a protein that cycles in many of our cells, called cry or cryptochrome, believe it or not, that protein was first discovered in plants and later discovered in humans. A beautiful example of natural science. This protein plays a key role in metabolic disorders, in inflammation, and in cancer. And we've begun to use genetics, the kind of genetics that I told you about, either epidemiological genetics like with 23andMe, or mouse genetics using CRISPR and other gene editing type techniques, that have really begun to tell us that as we modulate, something went wrong with my, sorry about that. As we modulate the level of this cog of the circadian clock, this core clock protein, we can have an impact, a beneficial impact in multiple different diseases. So the big question then comes in the last few minutes of my address is, how do we find specific drugs that alter circadian rhythms? What should we do? Should we take these beautiful lilies and make extracts from them and test if we can find compounds from them that would affect the circadian clock? That's certainly one kind of approach. But what we tend to do, of course, is to learn from what pharmaceutical companies have done. And we have large, diverse libraries of compounds. And what we need to test those large, diverse library of compounds is a surrogate or biomarker for the circadian clock. We can't take mice, for example. Mice have a beautiful circadian rhythm. Mice will go into a running wheel every night, and they will run in that wheel for about 5 miles. That's about 8 kilometers. So that is a beautiful circadian rhythm, but it's way too expensive to take 100,000 mice and inject each one with a different compound. We need a high throughput assay for this circadian rhythm, and that's exactly what we do, is we have developed human cell lines where we have CRISPR'd in the luciferase gene into the genome so that we can get these beautiful circadian rhythms and put them into a high throughput assay. So we can do a high throughput assay for chemicals that affect the circadian clock, and we have built a huge array of robotics using engineers who normally make cars. So engineers who make cars are generally much better than the engineers who make laboratory instruments. So if you want a real engineer to come and help you in your lab, don't get one who makes laboratory instruments, get one who makes automobiles, because they have much more stringent specifications than laboratory automation normally has. So working with engineers and bioinformaticians like that, we developed high-throughput screening to look for chemicals that specifically change our circadian clock. And of course, I wouldn't be telling you about it if we weren't successful. So what we actually discovered was chemicals that bind into a specific pocket in the cryptochrome protein. And what these chemicals do is to stabilize the cryptochrome protein. Probably about as much detail as you need to know for this afternoon. trying to listen to my talk in English. We are able in our lab to use some very fancy chemistry, and this is with collaborators that we have in Japan. I actually have a laboratory also in Nagoya in Japan. And we're able to use very fancy chemistry to improve the properties of those chemicals that we get in our screens. And now we can start to apply those chemicals to mouse or dog or other animal models of disease. And what do we find? What we find is when we use these chemicals to modify the circadian clock, we can get a benefit, for example, in diabetes by actually lowering glucose utilization. We can also sensitize the animals to insulin. We can show a benefit in something called HOMA-IR, which is a measurement of your metabolic fitness. So what I just want you to take away from this is in the 4th century BC, 2,000 years ago, a philosopher called Androsthenes worked for Alexander the Great. And of course, with the One Belt, One Road program of your government, the whole idea of trade routes that we're opening up is now very much in our minds. And what this philosopher did was he noticed every day that a plant called mimosa that grows in Asia would open its petals every morning just before the sun rose and would close its petals later in the day. And for the pleasure of Alexander the Great in the 4th century BC, he would write poems about how flowers would open and close at different times of day. So we've gone in just a short 2,000 years, we've gone from observing 24-hour rhythms in different organisms to a molecular understanding of those rhythms. And now we're in a position to be able to use modern genetics, genomics, high-throughput technologies to manipulate the circadian clock with drugs and to produce benefits for our patients. Now, I just want to leave you with one final thing, where this field is going is not only in the treatment of disease, but potentially in prevention. What one of my former students has shown is that if you take lab animals and you put them on a high-fat diet, what do you think happens to those animals? They turn into little balloons, little soccer balls. But if you only let those animals eat when they're active and not eat when they're inactive, they are resistant to a high-fat diet. So that is the idea that we are not only what we eat. So that's a famous saying in the West. I don't know if you have it also in China. But we are also when we eat. And we're now commencing studies in humans to really investigate the deleterious effect of eating at night or late at night, because that disrupts your normal 24-hour regulation of metabolism. And there could well be an advantage, for example, to shift workers, because we know, for example, that nurses have who are on night shift, have two major signals. They have a higher incidence of breast cancer, and they have a much higher incidence of obesity and diabetes. And we think that's all due to circadian disruption. So the next challenge for my field is not only to make drugs for circadian rhythms, not only to develop apps on the iPhone that help, or the Android, that help not to disrupt your sleep-wake cycle, but perhaps also to bring in a preventative philosophy where, coming back to the original idea of chi cycles, the better we harmonize with our 24-hour environment, the more healthy we may live a life. I apologize that I have no Mandarin and that you had to listen to this in English. I hope you learned a little bit today. I'm certainly learning a lot from the other speakers, and I thank you for your attention.